I am going to start with the electron transport chain now. Important part of the electron transport chain is that it is going to be generating some reduced coenzymes that we'll use for making energy. When we're in glycolysis, we're actually making a little bit of ATP, but really the NADH, right? 2 NADHs is equivalent to 5 ATPs. So we get a lot more out of these 2 NADHs than we actually get from generating ATP during this process. So again, each of these, this is a 2-to-5 ratio here, and that's the important part. This is where we get most of our energy, is from our reduced coenzymes, actually. As you can see here, we get 5 at the beginning of glycolysis. From pyruvate to acetyl-CoA, we get another 5 ATP equivalents. And then during the citric acid cycle, we get NADH, we get 6 reduced coenzymes that's equivalent to 15 ATPs, and then these 2 FADH2s, which is equivalent to only 3 ATPs. So not as much energy in these reduced coenzymes than in NADH. The citric acid cycle process is happening in the mitochondria, bringing in these 2 carbons from acetyl-CoA, these acetyl carbons, and these are then becoming completely oxidized, and they're being released as carbon dioxide here, and then we get a lot of reduced coenzyme out of this as we spin around this cycle. Kind of the net output here is that the acetyl-CoA, we end up with 2 carbon dioxides and reduced coenzyme A with these hydrogen ions that are going to be stored on one side of the mitochondrial membrane, and then a bunch of electrons that we get out of this, which are going to be transferred from one molecule to the next down this chain. This is just kind of a diagram to kind of show us where and follow those electrons. What do you mean by how do we get 8 electrons out? When we have acetyl-CoA, we have, these are all of the shared electrons in this acetyl group that are attached to these compounds. And you can see each of these has 8 different electrons that are in the valence shell. When we have water and we have a hydroxide ion. When we make carbon dioxide, we basically get, right, this is what we're ending up transferring this acetyl group and the water. We actually get the same amount of carbons and hydrogens and oxygens in this whole complex here, but we actually release 8 electrons from these groups, and these 8 electrons are going to different compounds. So they're being, we're losing electrons from this system, and these are going to be allowed to be used to generate more energy. Also, this hydrogen ion is going to be a really important component of this process as well. This is the mitochondria, and here is just an electron micrograph of this. The mammalian liver contains up to 5,000 mitochondria in each cell. So we have a lot of these things. These are the workhorse of the cell and generate a ton of ATP for our body. And here we have the citric acid cycle is happening on the inside here. Succinate, fumarate, NADH, NAD+. These are components of the citric acid cycle down here. So as reduced products such as NADH and FADH are oxidized back, so as you look at these complex 1, 2, 3, and 4, they're reduced through here, and they as they go through this process, they're pumping those extra hydrogen ions that I talked about earlier into this intermembrane space. So this is between the cytosolic region and the mitochondrial matrix on the inside where the citric acid cycle is happening. And we'll talk about that process and how that generates energy in a second here. The process of pushing these hydrogen ions on this side creates actually a gradient. It organizes hydrogen ions on one side of a gradient. So we're going to build up potential energy here that's going to then allow us to use that energy to synthesize ATP in this last step. So the electrons combined with oxygen in this end step here, one of the last steps, to form water, and that helps to pump hydrogen ions, and then these hydrogen ions can flow through this ATP synthase. It's an ion channel, but it's being driven by this concentration of hydrogen ions. Here we can see that in the electron transport chain, the number of electrons that are transferred is defined by this N here. So looking at Faraday's constant, and the number of electrons, and that's the change in energy. So if we can convert voltage into energy, we can convert that from a voltage to a kilojoules per mole. So that tells us how much the voltage difference from the inside to the outside of the membrane. That gives us this change in the energy. So the potential energy and the final energy. So as these electrons flow, there's actually the free energy that's associated with those changes from complex 1, complex 2, complex 3, complex 4, right? That flow of energy, it becomes less and less as the electrons flow through. And as we're doing that, as we're flowing through this pathway, we're hopping from one complex to the next. We're actually we're using some of that energy to transfer those hydrogens across the membrane to build up that concentrated hydrogen ion. So as you can see, the oxidation of each of these complexes, 1, 2, 3, and 4, the Gibbs free energy is negative. So this is going to happen. This is a spontaneous reaction. The oxidation of these complexes releases energy, and so that's important. That allows the electron transport chain to happen and to release small bits of energy as this complex goes through, and part of that's by the transport of the hydrogen across the membrane. So we have these complexes are on the inner mitochondrial membrane space between the mitochondrial matrix and the inner membrane space, and this would be the cytosolic region of our cells. Here's just a kind of zoomed in vision of complex 1, the NADH ubiquinone oxidoreductase. This is the chemical reaction that's happening. As the NADH comes in here, it actually is going to be oxidized, and it itself is going to reduce the FMN complex. And FMN is going to then transfer the electrons to the iron sulfur complex, and then transfer that into this quinone is reduced with those electrons as well. Quinone is going to be something that's going to be in the membrane. It's going to be able to move within the membrane and interact with other complexes. So this is helping with the proton motive force. That's another kind of term. The proton motive force is during this process, this is going to take 4 protons from the matrix and transfer it into the inner mitochondrial space. This is just the structure, kind of showing you that picture of what it actually looks like, where we have the FAD is here, and you can see there's close treatment of these. We have the FAD, and then it bounces to the iron clusters, can bounce to the ubiquinone and the heme center. These are all within the membrane, and these are all kind of happening, and they're all attached and part of that complex. So again, that QH2 right here, reduced ubiquinone, that's associated with the membrane, that can come in and out of this protein complex and flow through that membrane. So we get to complex 2, where we have this the same kind of thing. We have a Q. The ubiquinone is associated with the membrane, can kind of come in and interact with this complex. This is another complex where we can feed in. This actually feeds in kind of separately. It's a separate pathway from complex 1. This is where it uses succinate and fumarate, part of the citric acid cycle, to directly reduce the FADH2 coenzyme. So we get a little less energy out of this than we would if we were making NADH, but it still works. We still transfer electrons from this reduced coenzyme to the iron, that then transfers those to the ubiquinone. So it works separately from complex 1, but it still reduces ubiquinone in the end. And here is the complex structure 2. You can kind of see it's a dimer here. You can see both sides of that coming together, and it's going to be the membrane is this side, and then this is the mitochondrial matrix side, and this is the inner membrane space. This is the iron clusters here, the heme as well, that is going to interact and help transport these electrons through and generate this reduced ubiquinone. Complex 3 is where we have the interaction of now the reduced ubiquinone compounds pass electrons. They travel in 2 separate pathways here, and it provides, again, proton motive force. So this is really important. So we're going to get protons, you can see here, The ubiquinone is going to be traveling in the membrane. It's going to interact with this. It's going to transfer electrons, so they can come in this way and transfer electrons through these iron clusters to the different cytochrome complexes. But we're also going to be moving hydrogen ions. They can also come in through this region as well to reduce coenzymes as well, ubiquinone. And so again, this is going to move protons. We're going to move electrons in these different pathways. And this is the complex structure 3. And you can kind of see the change between the reduced form and the oxidized form. That's going to actually change the structure of this complex and the function. And so this is going to allow us to, these electrons kind of bounce from these different hemes and these iron clusters through this complex to transfer electrons. And you just kind of note this small structural change, especially of this alpha helix, as it changes over and moves from the oxidized versus the reduced form. Once we have this cytochrome C reduced, it can travel to complex 4. Here the heme groups and the copper atoms are reduced as electrons flow to the oxygen. So we have this reduced compound. It's going to release those electrons and become oxidized, and this copper then inside is going to be reduced, and we're going to travel all the way to this A subunit, another copper subunit, and then the electrons are finally going to be passed on to these oxygens and the hydrogen ions to form water, to form H2O. So this is why we need to breathe oxygen, because this is the final electron acceptor, where these electrons have traveled all the way through this electron transport chain to oxygen to form this water molecule on the inside of the mitochondrial membrane. We also have a proton motive force, so we're moving protons during this process from one side to the other. So you'll notice we've moved a lot. We've moved those electrons, but we've also moved a lot of protons as well. So again, if we don't have oxygen, if we aren't breathing, we don't have final acceptors for this process, and this halts the entire chain. It slows everything down. It halts all of this. It halts all of the ATP synthesis. So that's why we need to breathe constantly. So we have final proton or electron acceptors to make water, and to keep pumping these hydrogen ions on this side of the membrane. For this final step I'll talk about in just a second. So you'll notice that there are a few hemes. This is complex 4 structure. You'll notice these hemes within the protein, but then we've pulled them out here. And these hemes actually have this copper bound. So copper is really important in our diet, to have a little bit of copper, obviously, for our electron transport chain and some of these hemes. A lot of times you'll notice hemes have these centers where they're going to be bound to different types of metals that are important for the properties of those enzymes or proteins that they're associated with. So this is important for electron acceptance and the electrons flowing through these copper ions that are held in our membrane-bound proteins. Here's another electron micrograph of the mitochondrial inner membrane, and this is the complex 5. This is the ATP synthase. So these are, each individual one of these is a little ATP synthase component grouping of proteins, and I'm going to show you the cartoon version of this. So I have a lot of alpha helices. It's membrane bound, so it's attached to the membrane, and then it's got this kind of cap at the end. We're going to look at what that does and how this all works. The protons that we've moved to one side of the membrane are going to be used to now synthesize ATP. Here we're going to look at the concentration gradients that are set up during the citric acid cycle. And we're going to use ADP, an inorganic phosphate, to create ATP. And it involves the ATP synthase that's going to be found in the membrane of the cell. Here's the mitochondria, and we're going to see the ATP synthase here. So a lot of hydrogen atoms on the top end here. We're looking at the hydrogen atoms up on here, and those are in a lot more abundance than they are on the matrix side. You can see here that the hydrogen ions attach to the ATP synthase, and then it actually transfers hydrogen atoms to the matrix space. And when that comes in, that actually rotates in, that brings in another hydrogen, and that actually causes a turn on this protein. So we can see the protons entering here, causes the turn. We have hydrogen leaving, and we have the spinning of this bottom part that brings in enough energy to eventually activate the synthesis of the ATP compound. There's another video that I want to show you quick. There's actually, you can see the rotating of this gamma subunit. So what these scientists did is they took the beta subunits and attached them to a cover slip, a glass cover slip. And then they actually put on an actin filament that they labeled, and they were able to kind of bind to this gamma subunit, and then it would spin around, and you can actually see that as the protons flow through this channel and turn this subunit. So let's look at this for a second. So you can see that these are the actin kind of filaments kind of spinning around. So these beads were attached to the subunit, and then you can actually see that spinning around under a light microscope. We're done at Oxford University, but pretty cool that we can actually see those rotating in real time with a light microscope. So again, what we saw spinning there was this gamma subunit was spinning as when this was given protons that were then able to flow through here and spin that top part. So this is really what the energy that's provided to generate that ATP that we see is synthesized. You can kind of see that there are different levels of binding of these subunits that are around these alpha and beta subunits that attach to the ATP. So we have the ATP that is ADP comes into the open construct when we have a turn. That turn of the gamma subunit actually changes the conformation. So this changes, and then it's bound tight, and it hydrolyzes this into ATP, and then it turns again, and then it's loose, and then so we have ADP and inorganic phosphate. So we can see this thing rotates around, and it changes the conformation, and changes the ability to actually hydrolyze things and add that ATP synthesis reaction. This process happens really quickly, and there's a lot of these in our mitochondria. So the average person is estimated to make 9 times 10 to the 20th molecules of ATP per second. That's nearly 1 sextillion ATP molecules per second. Pretty crazy. I thought this was kind of interesting. So to look at the mitochondrial genome, it's a pretty simple genome. There are some RNAs in there. There's the cytochrome components we were talking about in the electron transport chain. Complex 1 is in green here. These green genes are complex 1. The purple genes are complex 3. Pink genes are purple complex 3 is complex 4 is the pink genes. Yellow are complex 5. Dark pink are some tRNAs and amino acids. And then the D loop is highly variable, used to actually trace evolution of modern humans. This little teeny bit of DNA provides evidence that we are actually all descended from a common ancestor originating from Africa. So this is all of our mitochondrial DNA. It's coming from our mother. So our moms put in their eggs before we're born, before we're fertilized, puts all of our mitochondria in the egg, and so the sperm doesn't provide much mitochondrial components that actually make it into the final product of our bodies. So our mitochondria all come from our mothers, and so this is where we inherit that. It's pretty interesting. So the oxygen that we breathe in, more than 90% of that is actually used in the electron transport chain. Most of the other stuff that's happening outside of that is not necessarily good. That is really important for the electron transport chain, but some of it can turn into non-good other products. So superoxide, hydroxyl free radicals, and hydrogen peroxide, and things that can essentially break DNA and cause other problems in our cells. These different superoxide ions and free radicals can grab electrons from bonds other than being involved in this electron transport chain, such as DNA, which can break bonds and cause mutations in our bodies. That's where these excess oxygen from high oxygen concentrations in our bodies, inflammation, radiation, smog, aging, different kinds of drugs and chemicals, and some of these actually we should be kind of going away from O2. This is what can happen when we have too much. And these reactive oxygen species that I showed you on the last page can cause cell injury and kill cells and cause cancers and other issues. So the conditions that can enhance the production of these these offshoots, such as these aging and inflammation, those are some of the drawbacks that can be caused by environmental issues. So we can actually protect ourselves from these harmful oxygen species. We have an enzyme called superoxide dismutase, and I said H2O2 is a bad side product, but it's actually a lot better than this superoxide ion. Superoxide dismutase, or SOD, actually creates this hydrogen peroxide compound and catalyzed one of the most effective enzymes known. It's a nearly instantaneous reaction. Once it finds H2O2, almost instantaneously converts that into water and oxygen. So these enzymes help our bodies protect ourselves from these harmful reactive oxygen species. This is an example of, when we talk about antioxidants, what does that actually mean? It just means that these things can protect us from those harmful oxidizing compounds. That basically vitamin E, it can become an electron acceptor, and basically we can look and see. You can see here vitamin E. Here, this is a product of vitamin E reacting as an antioxidant. We can see that this becomes oxidized compared to over here. We can break this bond here, and we can attach this hydroxide. It can accept one of those oxygens, and it doesn't change this. It doesn't make this more harmful. It just kind of breaks this chain open. So things that are antioxidants, it just turns into a different product that's not harmful to our bodies that then can eliminate this, but it's kind of a sink or something that can absorb those dangerous free radicals and kind of disarm them by turning them into something, a non-harmful byproduct. So this becomes oxidized and it actually helps our bodies protect ourselves from those things. So that's why they're saying, well, these things have antioxidant properties. So vitamin E, for example, it's good to have in your body because it can soak up some of those excess free radicals in that way. We can also, there are techniques for looking at the metabolic activity of different cells in our bodies, or if you're using cell culture, you can look at the load of hydrogen ions that are available or released through lactate. So we have different ways of measuring anaerobic and aerobic activity through the different oxygen consumption rate, or the OCR, or the extracellular acidification rate. So remember, those hydrogen ions that are kind of in this membrane, they can, depending on how much lactates there is, that also lactic acid, right? It changes the acid concentration of how the cells are. So we can actually measure those things with different instrumentation. There's this instrument called the Seahorse. It can actually measure this oxygen consumption rate over time, and you can kind of poke holes and essentially mess up, add inhibitors or activators of these different things. So we can look at oxygen consumption rate, and we can add different things to block, for example, the ATP synthesis that blocks the ATP-linked respiration. We can look at the permeabilizing the membrane and looking at how that influences, right? We know that hydrogen gradient is really important. Essentially eliminate the electron transport chain gradient because that hydrogen gradient's affected. So that's going to look at changes there. And then the mitochondrial inhibitors. So it will block the electron transport and mitochondrial related respiration before we get to the consumption of oxygen and what that does. So it should that's why you see this. The oxygen consumption rate should go down a lot, right? So it should go down a lot if we're blocking this third step where we actually create water from or right before oxygen is consumed here. So we're actually looking at just non-mitochondrial respiration, anything that's not involved there. So you can see when we were talking about that's about 10% of this total consumption of oxygen. There are ways that we can look at cellular health and cellular respiration on just an individual in cell culture. And there's just these are some raw data. I'm not going to go into too much detail, but you can look at, the baseline versus the ATP linked respiration, maximal respiration, proton leak, and you can look for differences for healthy cells versus sick cells. And so it's just a way that people can look at, maybe someone is looking at heart disease or liver disease. If the cells are not respiring properly or they're not regulating things properly, there might be a mitochondrial issue that's causing sickness and diseases. That's all I'm going to talk about for the electron transport chain. Hopefully you found some of this information interesting, and that is the end of this section here.